Okay, so a very good evening to. Oh, sorry, it's not good morning, it's good evening. So, yeah, a very good evening to one and all present here. Welcome to the week 12 course on Introduction to Complex Biological Systems. Right, so today is basically the last week course, and after that, you guys would be having the exams, as I suppose. So, right, so I'm sharing my slides with you all. I guess my screen is visible to you all, and I am audible enough if that is so. Okay, so right, so this week's course, that is the last live session. So today I will be mostly talking about the biophysical and the biochemical methods that involves the principles and the practices that we regularly perform in our daily experiments. So starting with, we will be dealing with the introduction to proteins. So there are different ways of how you can isolate proteins. First is from the natural sources, and then there are different recombinant proteins that are expressed from the different sources. Like from the natural sources, that includes the whole organism. You have the tissues, embryos, tissue culture cells, the microorganisms, things like that. And from the recombinant proteins that are expressed from the different sources, it includes the microorganisms, the fungi, the plants. You have the mammalian cell systems, the whole animal production systems, insects, things like that. So protein purification, so the general strategy is basically how you are extracting the protein out of that particular cell in which you are growing. That can be a tissue or a microorganism that you're disrupting to undergo a crude fractionation followed by a selective fractionation to get only the protein of your interest. But what you get is not the protein of your interest, rather quite a crude extract, maybe a little bit of a filtered version, but more of a crude extract, which you have to go through a lot of purification steps to actually get the pure protein form. So protein isolation methods can include like the salting out, that is based on the solubility of the protein of interest. Then you can go with centrifugation based on how heavier the particles are and how heavier your protein of interest is from the other crude materials. And the third thing is chromatography, that you can perform a few different steps of chromatography based on size, mass. That includes the size exclusion chromatography, also known as the molecular sieve gel filtration chromatography. Um, yeah, so that includes the the size exclusion chromatography, also known as the molecular sieve gel filtration chromatography. If you want to separate from based on charge, you can perform an ion exchange chromatography. If you want to do it based on hydrophobicity, you have a hydrophobic interaction chromatography, the reverse phase chromatography. And affinity chromatography is a widely used technique for high purify to purify proteins with high purity. So if I go based on a selective precipitation, so it is entirely based on the ionic strength, pH, and the organic solvents that we are using. So if I think of a molecular crowding agents like the polyethylene glycol, so specific property of the target protein based on the charge or hydrophobicity, solubility, and size, you can actually selectively precipitate out the proteins, and you can get a purified protein of your interest, right? Now, the other form is a salt fractionation. So, very high salt concentrations, if you are using, that leads to salting out of the protein, whereas mild increase in salt concentrations leads to salting out. So, as you try to increase the salt, slowly the solubility of the protein increases, and there is a limit above which, if the amount of salt increases, then it leads to salting out, and that will lead to the precipitation of all the proteins of your interest that are not soluble in such an ionic condition. So mild increase in salt concentration leads to low ionic strength, so the solubility increases, leading to salting in. Whereas if you are increasing the salt concentration to such an extent, that leads to the decrease in the solubility as the ionic strength increases, and we call that as the salting out. So there are some ions that are more effective than others in affecting the protein solubility, and this ranking is known as the Hofmeister series. So Hofmeister series basically tells you that which combination of the cation or the anion is basically going to give you the best property, the best ionic condition for your protein of interest. However, there are certain ions that you might be aware of, like NH4 plus, that stabilizes the protein, but also at the same time with an increased amount leads to salting out. At the same time, if you go to guanidium, there is no salting out because the stability of the protein is not there. The protein gets unfolded, leading to disordered protein, and when you have a disordered protein, so all the entire condition in guanidium leads to a disordered protein formation, and that will help to solubilize the protein but not stabilize the protein. Similarly, denaturation of the protein can be done in the form of a guanidium. You can do it in the with thiocyanides. If you want to keep it natured, soluble, then you can use sulfates, phosphate buffers, acetates. These are good combinations that you can use. So what's the mode of action? So high concentration of small, highly charged ions such as the ammonium sulfate compete with the proteins to interact with the water molecules. Now water removed from the protein solubility decreases, resulting in the precipitation. The other very interesting technique that is widely used in any experimental labs, in any protein labs, or in most of these biological labs is a centrifuge. You can find a centrifuge in almost every lab that is doing research, because it's a very interesting instrument, a very important technique for protein purification. So what's a centrifuge? So centrifuge is that uses the centrifugal force to bring down the particles, so based on how heavy they are. Okay, so you have different types of centrifuges. That can be a fixed angle rotor centrifuge, that can be a swinging arm rotor centrifuge. So what it does is, you put it over there, there is a centrifugal force acting just perpendicular to the swinging, to the rotating axis, and then it leads to fractionation. So you have a cell homogenate. Before centrifugation, you centrifuge it, and then what you get, so you get a supernatant and a pellet. So the pellet is probably the larger and the more dense components, and the supernatant is the smaller and the less dense component. So centrifugation is separating based on density, how dense the particles are, how heavy they are. It's like that. So if you have a cell homogenate, a very crude cell homogenate, and you want to undergo a low speed centrifugation, so what do you get? You get the pellets in the form of a whole cell, nuclei, cytoskeleton, and the other half remains in the supernatant. Now the same supernatant is taken in another time, it's centrifuged at a higher speed. So if it's centrifuged at a higher speed, you get the pellet in the form of mitochondria, peroxisomes, and the lysosome. You increase the speed, and then you probably would be getting the microsomes and the small vesicles, and when you go for a much higher speed, you can extract the ribosomes, viruses, and the large macromolecules. So based on the component from the cell that you actually require, you can choose that speed. But generally is recommended, if you need something that is say a mitochondria or a lysosome or a peroxisome for any kinds of an experiment, you first need to give it a very slow centrifuge, such that you can eliminate out the whole cells, nuclei, and the cytoskeletons before you have started to extract the mitochondrial lysosomes and the peroxisomes. So that is how it is generally done, because otherwise it will lead to uh, unnecessary things coming up in the pellet, which might not be quite helpful when you are doing a experiment or extracting a particular cell organelle. So that's the general idea of what I'm trying to do. So you spin it, you get the different components at the different speed at which you are actually centrifuging. So I'm not going to all of that. So ultracentrifugation techniques leads to finer separation and less convective mixing using the density gradients. You can use sucrose, you can use cesium chloride, right? There are some sedimentation techniques like the velocity sedimentation and the equilibrium sedimentation. So what is sedimentation? So it uses the force, the centrifugal force, where it's dependent on the angular velocity, that's radians per second, the distance from the axis, and the molecular mass. It also takes the, it's related to the buoyancy, that is the effect on the displaced mass, the friction, that's the frictional coefficient, and the radial velocity, and obviously the diffusion that occurs throughout the time of the sedimentation process. So the diameter of a single hemoglobin molecule is around 5 nanomolar. So the protein molecules can be quite concentrated. For example, the highest physiological concentration of any protein are the crystallines in the Iceland cell, that can be above 50% protein by weight. And fibrinogen is a large rod-shaped molecule that forms a fibrin blood clot when activated. So that's how you can purify proteins. One such technique is that. But how do you do it so that's all from the natural sources. How can you do it recombinant, using some recombinant techniques? So if you understand that a protein is the translated form. So our DNA, you go to an mRNA and then to a protein, you know that. So protein means the final form, but we can actually recombinantly make using recombinant DNA technology. We can clone that particular insert coding for a particular mRNA that would be further translating into the particular protein. Okay, so you can take that DNA sequence and insert that in a vector. So the DNA to be cloned, and you have the expression vector DNA. So they can be using the cloning technique. So you can recombinantly produce a vector insert combination called a plasmid, and that plasmid is then transformed into a bacterial cell, a competent cell for the construct to survive, for that vector, for the plasmid to survive in that particular bacterial condition. And then you can use some expression conditions to express that DNA to a protein. So for the bacterial protein expression, you need appropriate growth conditions, and you need to harvest the cell, you need to lyse the cell, and then take out the protein of your interest. So there are certain expression vectors. Most of you must be aware of it. If you are thinking of a prokaryotic system, it can be a p-test pet vector. Then a eukaryotic expression vector can be yeast, virus, mammalian cells. If you think of the host system, then prokaryotic can be a bacteria, the eukaryotic can be insect cells, then the yeast cells, and the mammalian cells like the human embryonic kidney cells. So what do we do for a bacterial protein expression? So there is an overnight transformed bacterial culture which is inoculated freshly into LB medium. So LB is a medium where bacteria loves to grow. It's the food for an bacteria. So when the OD reaches 0.4 to 0.8, so why is this so? Because that is the time when it reaches the log phase. That's the time when the bacteria grows exponentially. So that is where it's the best time to inoculate, add IPTG. So IPTG is a inducer. So it's a molecular mimic of allolactose, which is a lactose metabolite. It does not take part in a metabolic pathway. It triggers the transcription of the lacoperon, and it's used to induce a protein expression. So upon the addition of IPTG, the protein expression starts. After maybe 12 hours, uh, then you can first, what you can do after induction of IPTG, you need to incubate it for depending on how fast your protein grows or how slow it grows. Depending on that, you can fix up a time and the temperature, and you can grow it for that specific time and temperature. And then what you need to do, you need to pellet down the cells, centrifuge the cells, pellet them down, and harvest them. So how can you purify the proteins? We discussed about the differential centrifugation, then differential salt precipitation, You can use differential solvent precipitation, preparative electrophoresis, and column chromatography. Right. So these are the different techniques of how you can purify the proteins. So differential centrifugation is like the way I discussed over there. It's separation of proteins or any material on the basis of their size, mass, and density. It's a function of the size of the protein and the speed if centrifugation, and it gives us a rough separation. It's basically fractioning. So like first we are separating out a few bunch of organelles, then we increase the speed of the centrifugation, and then we are eliminating a few others, and so it's a differential, means you are increasing the speed of the centrifugation slowly. The other way out is dialysis. So it's based on an osmotic pressure. It allows you to get rid of most salt ions, and it is impossible to remove the salt completely. It is because it's always in an equilibrium. So when the equilibrium condition is reached, it's the exchange stops. So therefore, it is always recommended that you should have at least 20 to say at least 100 times more amount of buffer than what is the amount you are having in that dialysis bag. So that is why the salt concentration decreases 100 times, which is actually quite negligible considering the amount of salts that or the contaminants that are present in that crude extract that you are actually dialyzing. Column chromatography, it's the most and the best approach for purifying large amounts of proteins. It achieves highest level of purity and largest amount and requires low effort. The lowest likelihood to damage the protein products. So standard method, and it's a standard method for pharmaceutical industry as well. So what are the techniques? You have a gel filtration, ion exchange, and an affinity. So in short, gel filtration is how you are separating them based on their size. So it's a separation based on size. So protein molecules separate based by size. So larger molecules pass more freely, appearing in the earlier fraction. So they elute faster than the small molecules because there are certain pores. So the small molecules can enter even the bigger pores and the smaller pores, but the bigger molecules cannot enter the smaller pores. So that is why they elute faster than the smaller protein molecules or the smaller molecules that you have. Ion exchange is that you're basically you're purifying them based on their pi. So what is pi? So pi is the isoelectric point. So every protein has an isoelectric point, the point at which it exists as a zwitterionic form. We have heard of it, but generally when the buffer condition is more than the pi of a particular protein, if the pH is of a buffer is more than that of the pi, the protein is negatively charged. If it's less than the pi, it is positively charged, and then you can use the oppositely charged ionic column for actually capturing it and letting the all others get rid. Okay, so that is how you, based on the pi and the charge of that particular protein having at the particular pH of a buffer, you can actually perform an ion exchange chromatography. And what is an affinity chromatography? It's because of the force of attraction. So for example, you have the nickel NTA bead. So nickel has an affinity for the histidine because it forms an octahedral geometry when it binds to histidine. So that is why, and it also has a better affinity for that. You can use even a GST column that also has binding affinity. So it's having an affinity for a particular tag or a particular stretch of sequences allows you to actually get a more purified protein, because that stretch would not be present in other contaminants or other proteins that are not of your interest. So that's how you are purifying them based on affinity. Okay. So maybe a little bit in detail. So this gel filtration chromatography is a size exclusion chromatography. So molecules are separated according to the differences in the size as they pass through a hydrophilic polymer. So the polymer beads composed of cross-linked dextran, which is light, highly and uniformly porous, like a Swiss cheese. So large proteins comes out first, small proteins comes out last. So we need to have a way where your protein is. So we need to have a standard or a molecular weight marker, add a target to your protein which you want to purify, an enzyme, add a substrate to the tubes to see where does it appear, and you collect it. So it's separating based on the size. The ion exchange, the principle is to separate on the basis of charge absorption. It has also has a highest resolving power, highest loading capacity, and it's a widespread applicability. So most frequent chromatographic technique for protein purification. Also, very important, if the pi of your protein is very high or very low, maybe say in the range of 9 or maybe 9.5, 10, then you can use a pH of buffer 9 or a buffer of pH 8.5 and purify your protein quite easily. Even if the pi of your protein is around 4, 3.5, 5, that is also quite applicable, because generally the bacterial proteins have a pi ranging from 6.5 to 8 usually, right? And affinity chromatography is the adsorptive separation in which the molecules to be purified specifically and reversibly binds to a complementary binding sequence immobilized on an insoluble support. So purification is 1,000 times better from a single step, and it's the preferred method if it's possible. So the protein is not covalently linked to the ligand, like you do not have the his tag or the stretch of 6 histidines bound to the nickel nickel ion quite covalently, because you can have another affinity towards the nickel that might be tighter than that of the 6 histidines, and that can easily replace it. And that is how we are actually purifying the protein. Then we can go for an isoelectric focusing. So isoelectric focusing is separating the proteins based on their charge. So it's based on the pi. Okay, so if you have different proteins over there, and when the proteins are actually neutrally charged, you will see them concentrating within the center. If it is of negatively charged, it will move towards the positive end, and if it's of positive charge, it will move towards the uh, negative end. So the other technique that we can use is a polyacrylamide gel electrophoresis. So it's a type of an electrophoresis where we run the proteins in a gel in an electrophoresis system. Okay, through constant current or constant voltage, you can tell, and there also you are separating them based on their resolving them based on their size and the shape. Okay, so basically the size, because the shape does not remain. Why am I saying it? Because if you're doing a sodium dodecyl SDS page or a sodium dodecyl polyacrylamide gel electrophoresis, there all the amino acids are negatively, uniformly negative charged, and that is why they are separated just based on their size. And when you stain them, followed by destaining, you see quite thick bands of overexpression of proteins or purified proteins, but there are no other contaminants in that particular lane. So preparative electrophoresis or a 2D electrophoresis is basically a combination of isoelectric focusing and SDS pitch. So what you first do, first separate the proteins based on their PI. Okay, so there will be different proteins are having different PIs, and you can separate them based on their PI. So you might be knowing, okay, so the protein of PI pH 3 or having a PI 3 comes over here, and then you're separating them, and then you turn this gel or whatever you have, a membrane or whatever, so you turn it and allow it to run it in an SDS page. Then you will see each lane that particular protein is coming in which lane, you can actually understand. So now you know. So in one axis you have the pH control, and in one axis you have the molecular weight marker. Okay, so the molecular weight, so the protein separating over here, so you know that this is my protein. So you are aware that the pi of your protein is 7, so you will be looking to that particular range, and you will tell, okay, so the protein that I'm looking for is this particular protein over here. So this is a 2D electrophoresis, basically, where you are separating first on isoelectric focusing, that is based on the pi of that particular protein, and secondly you are performing a polyacrylamide gel electrophoresis. Okay, Any questions till this much? Any questions? Because then we will go into the details of the electrophoresis system. Any questions till this much? Maybe we can take 2 minutes to think if you have any questions.
No, sir.
No questions? Okay. Let, okay. Maybe we can continue in 2 minutes. Okay, so we can go then. If you don't have question, then we can go move on to the details of the gel electrophoresis. So, gel electrophoresis is a technique used for separation of DNA, RNA, protein molecules using electric current applied to a gel matrix. Electrophoresis refers to the EMF or the electromotive force used to move the molecules through the gel matrix. Now, by placing the molecules in wells in a gel and applying an electric current, the molecules will move through the matrix at different rates, determined largely by their mass when the charge to mass ratio of all the species is uniform. So that's the principle of electrophoresis, where it uses the Ohm's law: voltage is equal to current into resistance. So gel can be viewed as a resistor and the power supply as the voltage and the current source. So most power supplies deliver constant current or a constant source. So some will deliver constant power, that's power is equal to voltage into current. So there is a modern equipment that's color coded so that the red is the positive terminal and black is the negative terminal. So this configuration is designed to work with a vertical slab gel electrophoresis in which the negatively charged proteins or nucleic acids moves towards the positive electrode in the lower buffer chamber, that's the anionic system. Now if you think of a single gel that's attached to a power supply in an anionic system, the negative charge flows from the negative cathode terminal into the upper buffer chamber through the gel and then into the lower buffer chamber, which is connected to the positive anode or the red terminal to complete the circuit. Now, pH is an ionic system based on the negatively charged HDS. Now, occasionally proteins are separated in cationic systems. Now, in these gels, the proteins are positively charged because they are very high pH and the gel buffers or the presence of a cationic detergent. Now, proteins move towards the negative electrode and the polarity is reversed compared to the HDS pH. So if you look at agarose, it is extracted from a seaweed and is a linear polymer of sugar molecules. Now, when heated in water, the agarose polymers are flexible and the mixture of agarose and water liquid. Now, when the agarose and the water cools, the agarose polymers forms a matrix. Now, when this happens, the mixture becomes more solid and gel-like, and the concentration of the agarose determines how dense the matrix is. And the concentration of the agarose also determines how well you want to resolve and what you want to resolve. If you're using a higher amount of agarose, say generally it's used a 1%. So if you're using a 1% agarose to make a gel, then that will basically lead to separation of small or lower order of the lower the base pairs or the lower DNA samples, but uh, DNA constructs. Okay, and if you're using a lower concentration of the agarose, then that will lead to the separation of usually higher base pairs. Okay, so this is how the agarose gel apparatus looks usually. So the you load the samples in these wells and you allow it to run at a constant voltage or current. So comparing the cut up DNA, so when you run a gel in an agarose apparatus, you see this is how the well looks like when you're taking an image under a say a chemiluminescent or some things like that. So how do we compare the different DNA fragments? So separate the fragments by the size. So if you're running a DNA ladder, so the DNA ladder will have the standards of different base pairs. Okay, so how do we separate the DNA fragments? So we run it on an agarose gel and then we perform electrophoresis. So the method of separating DNA is using an electric field. So DNA is negatively charged. So when it's in an electric field, it moves towards the positive side. So the fragments are basically swimming through the jello. So the smaller ones will migrate faster. The bigger ones will migrate slower. So DNA moves in an electric field. So how does you compare the DNA fragments? So the size of the DNA fragments affects how far it travels. So the smaller pieces travels farther, the larger pieces travel slower, and they lag behind. So DNA and the restriction enzyme. So you load them in a well and then you run it in a gel, and the longer fragments lags behind, the shorter fragments runs faster. Okay, so you cut the DNA with the restriction enzyme and then you run it in a gel to check whether your particular plasmid has been restriction digested or not. And if that is so, then it's well and good. If not, you have to repeat it. And that is how we basically do the recombinant DNA technology so far in this way. And then you can stain the DNA using Carolina blue stains, or if you're using some ethidium bromide, then that will also help you to visualize properly. So that's about the how you're separating the agarose or the DNA components. For a protein, we use a polyacrylamide sodium dodecyl polyacrylamide gel electrophoresis. This is how the apparatus looks. These are the different components that we use, and after running it in a gel and then when you stain it or distain it, it's basically this is the type of the gel that you usually obtain. So the lane 6 and lane 1 is the standard molecular weight markers that helps you to tell usually how the proteins of that particular size runs. So if your protein is overexpressed around here, you can tell, okay, so the protein size is around 97, maybe less than 97, 95 or something like that. That also you can calculate using log mw versus the migration rate, from where you can tell what is the exact molecular weight of that particular protein. Okay. Now coming to the separation, so we know that the gel is a matrix to separate the target molecule. So in most cases, the gel is a cross-linked polymer whose composition and porosity is chosen based on the specific weight and the target of the that we are wanting to analyze. Now, when separating larger nucleic acids, the preferred matrix is a purified agarose. So, but in SDS we use an acrylamide, which is in contrast to polyacrylamide, is a neurotoxin and must be handled with appropriate safety precaution to avoid poisoning. Now, agarose is composed of long unbranched chains of uncharged carbohydrates, and then we have some staining agents, some visualizing agents like the ethidium bromide, silver, or comacid blue dye, which may be used throughout the process. Now, if the analyte molecules fluorescence under the UV light, a photograph can be taken under the UV lighting condition. If the molecules contain radioactivity added for visuality, an autoradiogram can also be used. Now, what we need to observe is at which range your protein is actually being observed, and from the molecular weight marker that has been run on either of the lanes, that can be actually used to basically understand the size of the protein. So the distance a band travels is approximately inversely proportional to the logarithm of the size of the molecule, and from there, if you go for a thorough calculation, you can find out the actual size of that particular protein, right? Okay. Now, proteins, unlike the nucleic acids, can have varying charges and complex shapes. So, for proteins, we run a sodium dodecyl polyacrylamide gel electrophoresis. So, generally, the amount of SDS bound is relative to the size of the protein. That is usually 1.4 gram SDS per gram of protein. So that the resulting denatured protein have an overall negative charge, and all the proteins have a similar charge to mass ratio. Since denatured proteins act like long rods instead of having a complex tertiary shape, the rate at which the resulting SDS coated proteins migrate in the gel is relative only to its size and not its charge or shape. The native gel electrophoresis is basically a technique used mainly in protein electrophoresis where the proteins are not denatured and therefore separated based on the charge to mass ratio. Right? Polyacrylamide gel electrophoresis is used for separating proteins ranging in size from 5 to 200 kilodaltons due to uniform pore size provided by the polyacrylamide gel. Now, pore size is controlled by controlling the concentrations of acrylamide and bisacrylamide powder used in creating a gel. Acrylamide is a potent neurotoxin in its liquid and powdered form. The other types of gel used in agarose gel. So agarose gels can also be used to separate native protein. They do not have a uniform pore size but are optimal for electrophoresis of proteins that are larger than 200 kilodalton. Now, unlike SDS type electrophoresis, native gel electrophoresis does not use the charged denaturing agent. জানি প্লাস উপরে যেটা আছে ওটা তো আছে এমনি কটা রুম একটা yeah. So okay. So what is the basic procedure? So the solution of the proteins to be analyzed is first mixed with SDS. That's an ionic detergent which denatures the secondary and the non-disulfide linked tertiary structures, and it applies a negative charge to each protein in proportion to its mass. So without SDS, the different proteins with similar molecular weights would migrate uh, differently due to differences in the mass to charge ratio, as each protein is an isoelectric point and the molecular weight particularly to its primary structure. So this is a native page. So SDS binds in a ratio of approximately 1.4 grams SDS per 1 gram of the protein. Approximately uniform mass to charge for most proteins, so the distance of migration through the gel can be assumed to be directly related to only the size of the protein. So yeah, a little bit about the components that we are using. Mhm. So generally for an SDS stage, we use you can say agarose, acrylamide, polyacrylamide mixture. So based on the mixture and the percentage of C cross-link or the total acrylamide bisacrylamide monomer, we can actually determine the concentration of the SDS gel. So the pore size decreases with increasing percentage T with cross-linking. Say 5% gives the smallest pore size. Now upon any increase or decrease in the percentage C increases the pore size, as the pore size with respect to percentage C is a parabolic function. Now we can also use tris. So it has been used as a buffer because it's an innocuous substance to the most proteins, right? So its pK is 8.3 at around 20 degrees. So glycine, it's used as a source of a trailing ion or a slow ion because its pK is 9.69, and the mobility of the glycinate are such that the effective mobility can be set at a value below its slowest proteins of negative charge. So the other thing about it is that the acrylamide, which is a white crystalline powder, so while dissolving in water, autopolymerization of acrylamide takes place. It is a slow, spontaneous process by which the acrylamide molecules joins together by head-to-tail fashion. But in presence of the free radicals generating system, the acrylamide monomers are activated in a free radical state. So bisacrylamide is a cross-linking agent for polyacrylamide gels. Sodium dodecyl sulfate is a common dissociating agent used to denature native proteins to individual polypeptides. Now the detergent wraps around the polypeptide backbone, so the intrinsic charges of polypeptides becomes negligible when compared to the negative charges contributed by the SDS. So thus, polypeptides after treatment becomes a rod-like structure possessing a uniform charge density, that is the same net negative charge per unit length. Ammonium persulfate is an initiator for gel formation. Timid is used as a stimulator, right? It is initiated, it's an initiator as the APS, and then it's the catalytic, uh, timid is the catalyzer. Okay, so it can be initiated by the ammonium persulfate and the quaternary amines. The rate of polymerization and the properties of the resulting gel depends on the concentration of the APS and the timid that we are using. Now for visualizing, we need some chemicals for an SDS page. That's the bromophenol blue. It's known as a tracking dye. So when we are preparing the samples, we use the bromophenol blue as a tracking dye. So it's viable in alkali and neutral conditions. It can bind to proteins weakly and give it a blue color. Right, and if you want like to stain the proteins, which you would generally require, then you can use the Kumasi Brilliant Blue, which is the most popular protein stain. It's an anionic dye which binds to proteins non-specifically, and it's usually prepared in a non-polar solution of methanolic and acetic acid. So the proteins in the gel are fixed by acetic acid and simultaneously stained. The excess dye incorporated in the gel can be removed by destaining with the same solution but with a different type of a dye. Okay, so right. Any questions? Any questions till this stage? questions? If not, then we can start with the practice problems. So tell me, a protein will be positively charged when? When do you expect a protein to be positively charged? So when can a protein be positively charged? A protein will be positively charged when the pH is lower than the pi. So if the pH is higher, it will be negatively charged because it will be the pi means the pKs. So the pKs would be more shifted, so the hydrogens would be more lost. So right. So A is the correct option. pH less than pi. Now the next one, in size exclusion chromatography, proteins are separated based upon surface charge, size and shape, affinity, all of the above. So in size exclusion chromatography, how are proteins separated? Yeah, quite direct. And the correct answer is size and shape. So usually it's both actually. Why do I say shape? Because sometimes there are some proteins that form oligomers, so you can even see these oligomers in size exclusion chromatography. When they elute as oligomers only, right? So size and shape is the correct one. Now, which of the following statement is true? In SDS page, all proteins are negatively charged. In native page, proteins can be negatively charged, positively charged, or neutral. In agarose gel, nucleic acids are positively charged, and no denaturing agent is added in agarose gel electrophoresis. Which of the following statements is or not true? So there can be more than one option correct. So yeah. So in SDS page, all the proteins are negatively charged. That is right because you're using SDS. That's usually used for uniformly negatively charging the proteins. In native page, proteins can be charged based on their pI because it's a native page. You're not using any kind of additional agents. So A and B are correct. C and D are not correct. Okay. Which of the following statements is true? Polyacrylamide gel can be depolymerized by heating. An agarose gel can be depolymerized by heating. In the absence of bisacrylamide, acrylamide forms a linear polymer. It's easier to control protein size in polyacrylamide gels than agarose gels. Which of the following statements is true? Okay, so polyacrylamide gel can be depolymerized by heating. So heating is actually such a step that can depolymerize anything. So when it's an agarose gel or an acrylamide gel, so it does. So A and B are correct. In the absence of bisacrylamide, acrylamide forms a linear polymer. That is also correct. And D is not correct because we cannot. It's easier in a polyacrylamide than agarose. Not surely we can tell about that. So ABC is correct. Okay. Now, a mixture of three proteins of sizes 50, 30, and 12 kilodalton are separated by size exclusion chromatography. What is the order in which they will be eluting? So, the order of elution, is it 50, 30, 12, or all of them will come out together? Is it 12, 30, 50, or 30, 12, 50? So, in which order will they elute from a size exclusion chromatography? So in which order will they be eluting? C, 12, 13, and 50? No, it's just the opposite. The first, the higher molecular weight elutes out. Okay, so it's 50 first, then 30 and 12, right? Because the pore sizes, there are different pore sizes that are available, so the small proteins can enter the big pores as well as the small pores, but the big proteins can only enter the big ones, not the small ones, so they elute faster. So the correct option is A. You have expressed protein in bacteria and lysed the cells. How will you check if your protein of interest is present in the cell lysate? So how will you check whether your protein of interest is there in the cell lysate? So what is the experiment that you're going to do? Use DLS, SDS page, measure at 280, or a CD? Yeah, I've got one answer. A. Okay. So no, it's DLS. It tells you about the complexity of that particular protein. That's only possible when you have a pure protein. SDS page, definitely that is the only right way of doing it. Measuring at 280 won't give you the accurate data because there will be other proteins as well that will also contribute to that absorbance. And CD spectroscopy gives you about the secondary structures when it's a pure protein. Otherwise not. Okay. The next one is a protein without any affinity tag is being purified from sonicated crude lysate. Which of the following is the best purification sequence? So you have a protein without any affinity tag, which is being purified. So what is the best way you can, best sequence in which you can do it? So nickel in Ni2 plus column, ACC ion exchange, salting out ion exchange, nickel 2 plus column, ACC salting out ion exchange, ACC. I have got one answer. So if you look, the protein does not have any affinity tag, so I cannot use any affinity column or an affinity chromatography for doing it. So anyways, A and C are eliminated. So first you go for an SEC with the crude extract makes no sense because there will be so many other proteins as well that will also be contributing or maybe of the same size as well. So not first SEC. So you can first do a salting out that will eliminate most of the proteins in the first go. Then ion exchange because most of the bacterial proteins will be salted out, and then an ACC, because ACC you should be loading very close to a purified product to get an accurate size as well as purified proteins when you are sure that there is not very close around proteins in that particular extract. Okay, the next one. This is a equation. So, what is the concentration of the protein? So, you're given the absorbance, you're given the molar extinction coefficient, and you can consider the path length as 1 centimeter. So, what's the concentration of the protein? So, it will be A equals to epsilon Cl. Absorbance equals to epsilon Cl. So, you know absorbance, that's 9 by 11460, and that is the value that you will be getting in molar. I don't have a calculator. If you say that it is no, maybe 9 by 11 is uh, 0.79, and that's 1000 times, so maybe yeah, 0.79 millimolar. That might be correct. I don't have a calculator, so the concentration should be in millimolar, I guess, right? So yeah, possible 0.79 millimolar. Okay. Now, so the calculation is clear to you, I guess. Absorbance equals to epsilon Cl. We are considering we have the epsilon value and we have the path length as 1 centimeter. Okay. Now, which of the following can be determined using SDS page? Molecular weight of a protein, PI of a protein, presence of disulfide bonds in a protein, dissociation constant of a protein ligand complex. So, which of the following can you determine by an SDS page? So what does SDS page do? Basically, you have to think from that perspective. you're saying D. Okay, so the molecular weight of the protein can be detected using an SDS page. The PI of the protein cannot be detected. Presence of disulfide bonds in a protein can also be detected if you're not using any reducing agent like BME, DTT, things like that, or TSEP. If you're not using, it should be able to be determined in an SDS page, the disulfide bonds. And D, not possible. Dissociation constant cannot be determined at all. So A and C is correct. And I guess the last one, what is the role of SDS in SDS page? It acts as a reducing agent, imparts uniform negative charge to proteins, uh, controls the pore size of gel matrix, and determines the pH of the gel. Yeah, actually, that's correct. It imparts uniform negative charge to the proteins because we were studying around 1.4 grams of SDS is required to uniform negative charge the 1 gram of protein. So that's all. Yeah. So that's all from my part about this lecture. If you have any questions, any doubts from any of the previous lectures, you go ahead, ask me, because this is the basically the last class, and after that you will be having the exams. Those who are taking up the exams need to take these lectures very seriously. Everything is there in YouTube. The links are shared with you, the slides are shared with you, so should not be a big problem. So you should go through these lectures, the practice problems, very similar kinds of problems will be there in the exams. So yeah, you have to just keep practicing them, going through the lectures, the actual lectures, the brief lectures, the live sessions. Okay. So yeah, anything, any questions, any doubts? No doubts. Fantastic then. Yeah, best of luck for your exams and your future career. Uh, Probably we will meet again in the next year when I'm taking this the live sessions again, but with or maybe in some other lectures. Yeah, it was good interacting with you guys, and that's all. Thank you very much for joining and taking up this course. Best of luck for if you're taking up the exam, things like that. Yeah, good luck. Yeah, thank you very much. Thank you. Yeah, it's my pleasure. Thank you.